Hello everyone and welcome to this video. My name is Albert Wijngaard and in this video I will show you an example on how to use the new Myra Workbench software in a service lab setting. So let's assume that you operate a service lab in which you use both the MIC and a Bio-Rad CFX. Among your customers there are three hospitals, Hospital A, B and C, and you offer viral testing to these hospitals. So the viral tests offered are EBV, HPV and CMV. From each hospital you will receive request forms with patient barcodes and the tests for each patient. So some patients need to be tested on all viruses and others only on one or two. The next step is to merge these request forms into a work list, thereby making sure you put all the requested tests in one column and each test is separated by a comma, as you can see here. Once you have the work list ready, you need to save this as a comma delimited CSV file and you are ready to go. Although the example given here is for a service lab, it can be used in any hospital lab as well, where you have, for example, 3 doctors requesting any viral test for their patients. So programming the lab workflow is as easy as 1, 2, 3. The first step is to make your assays. Secondly, import your work list. And finally, add the components to your work deck and start the run. So make your assays by clicking on the qPCR assay icon. Now the good thing is that these assays only need to be made once. Once you've made the assays, you select Sample Prep and qPCR Setup. So let's start by making our first assay. So click on qPCR assay and select the chemistry type. So we would like to use hydrolysis probes. And we have a target. And the first target is EBV. And EBV is measured in the green channel. And we also have an EBV internal amplification control, which is measured in the yellow channel. Below here you will see the reaction setup for the Myra, and it has already added forward, reverse and probes to this reaction setup. But we have our own primer probe mix, so we are not going to include those. So we're going to untick this box for EBV and also for the internal amplification control. The next thing is to define our volume, 20 microliters. And we have water source template, but we also have our 2x buffer. And we have enzyme mix. And we also have our EBV primers and probes. So the next thing is to assign the tube types. So my water comes in a 5 ml screw cap tube and my 2x buffer is in a self-standing 1.5 ml tube, and perhaps my enzyme mix is in a 2 ml tube, self-standing, and my primers and probes are in a 1.5 ml self-standing tube. The next thing is to define the volumes. So water is calculated automatically. Let's say that we want to add 3 microliter of our sample. So this is the 2x buffer, so that needs to be 10, and our enzyme mix might be something like 0.25. And our EV primers and probes are 1 microliter. We also have controls. We have the NTC, and we're going to define that as an NTC, and that comes in a tube, a 1.5 ml tube. And we also have our EBV positive control. So this is our positive control, and that also comes in a 1.5 ml tube. And we also have an EBV negative control. So this is our negative control, and that also comes in a 1.5 ml tube. So for this part you are now done. This is the profile. I would say leave it as is, and you can add an analysis. So let's say that we want to add identifier to this. But in principle you're ready for EBV. So we're going to save this assay as EBV. If we want to make an assay for CMV, it's very simple. We're going to change EBV to CMV, like this. And we're going to do the same here. And of course here as well. And here also. And we do a save as. So this is now CMV. And we're going to do the same for HPV. So now we're done with making the assays, and the next step is to do the qPCR setup. So click on sample prep and qPCR setup. So the first thing is that we are going to load our assays. So we're going to select CMV, EBV and HPV. So now that I added my assays, I'm going to import my samples, and my samples are on a plate. So I select plate, and here is an icon that will allow me to import my samples. So this is the sample list I made previously. That is the CSV file. I'm going to open that CSV file, and what I will see is that this is my CMV file with the column headers. And I'm going to ignore the first rows because those are my column headers. So I'm going to do it like this, ignore the first row. Then I'm going to select sample name because these are my sample names, these are my barcodes. And these tests requested are my assays. So I'm going to select the assays. And here I will select groups. I can import that, but I can also save this as a template, so I don't need to define the column headers and the columns the next time. Save that and click OK. So it has now added the barcode names and the assays. The next step is to go to the reactions. If I click on reactions, you will see that the setup here is for a MIC rack. And I have my NTCs, my positive controls, my negative controls for CMV, but also for EBV and HPV. And I can now see that this sample here needs to go with EBV, HPV and CMV. So color coding is done automatically, and if I would like to have all these in duplicates, I can even do that by clicking here and just making it 2. There's also an option to say, look, I want to have everything in a 96 well plate for my CFX. Here. Okay. Well, that's how you change that. The filling order, either vertically, or maybe I want to have it horizontally. If we go to the deck layout, we will see that our samples are here, and this is my target plate. So the next thing is that I need to add my components. But before I do that, I would like to have my Myra 36 well block here. And I'm going to add my components. So I'm going to add water, 2x buffer, enzyme mix, CMV, EBV, HPV primers and probes, and I'm going to make my master mixes here. So this is the CMV master mix, EBV and HPV. So the total volume will be around 350, so I'm going to select these tubes, do a right click, and I'm going to change the selected tubes to 0.5 ml. There are also some samples here, so I can put my NTC here, my CMV positive control, negative control here, EBV positive negative control, HPV positive negative control. So this is all that you need to do. It will take around 19 minutes to complete. And the other nice thing is that you can even export your reactions to a CFX format, or to a Quant Studio format, or even to a simple CSV file. I would like to thank you for your attention. If you have any further questions, please do not hesitate to contact me.